Good morning, good afternoon, and good evening, everyone, from wherever you're joining us, and welcome to our first webinar in the Women's Health 2024 series. Today's webinar is titled Sex Differences in Risk and Resilience to Alzheimer's Disease, the Role of Genes and Hormones, featuring Dr. Rachel Buckley, Associate Professor of Neurology at Harvard Medical School and Massachusetts General Hospital. Today, she'll present to us the sex differences in dementia, focusing on diagnosis, tauopathy, and implications for clinical trials. Just before we get started, we would like to acknowledge our partners for the series, the American Physiological Society, the Society for Women's Health Research, and Scientist.com. Specifically, we'd like to give our thanks today to today's host society, the Society for Women's Health Research, for partnering with us on Dr. Buckley's event. We'd also like to say thank you and give a huge shout out to all of our series sponsors. None of this would be possible without them. I'm Sydney Otamanchak from the events team at Scientist.com, and I'm happy to be your host for today's event. Before I get started, I wanted to cover a few housekeeping notes to help you all get the most out of your experience today. First, you can have a look at the interactivity poll that's at the right of your screen with 4 tabs: chat, Q&A, polls, and handouts. This is where you'll participate in the Q&A during the webinar, as well as access all webinar resources. We want this to be an interactive experience for you, so please send in your questions, thoughts, and comments using the Q&A tab, and we will try to get to as many of your questions as we can at the end of the webinar today. When a question is added into the Q&A panel, you can upvote that question to increase its position on my list by clicking a thumbs up button underneath it. This is always helpful so I know which questions to address first. You can also explore some links and documents in the handouts tab to learn more. Lastly, if you're having any technical issues, please first try refreshing your browser and ensure you're using Google Chrome to access this event, as it is the most stable browser. If you're still having trouble after you've refreshed, you can use the private chat function to communicate with me by sending in a chat, and I'll help you to troubleshoot. And now I'm very excited to welcome Dr. Buckley to join me. Good morning. Take it
Good
away.
morning. Thank you so much, Sydney. And thank you so much to the organisers for giving me the opportunity to present. I'm the first speaker in the series. It's such an honour. And I'm here to present and really talk about sex differences in risk and resilience for Alzheimer's disease. My focus is primarily on sex differences, but I have a very strong slant and a very great passion for women's health. So I'll be speaking a lot about that, excuse me. So, I'd like to start out a little bit with some of the sort of basic principles of why we think about sex differences in risk for Alzheimer's disease. Women are at greater risk of dementia. So, if you think of all patients with Alzheimer's disease dementia diagnosed currently in the United States, two-thirds of those are women. And in fact, it's not just represented in those who are currently alive. When we think about mortality rates as well, women are more likely to die with a cause of dementia, whether it be primary or secondary, particularly past the age of 80. And we know that from some of the work that we have done in Australian death certificates that were captured for every death between the years of 2006 to 2014. And by and large, we see about a doubling risk of mortality with dementia, particularly past the age of 80. But it's not just about thinking of sort of prevalence rates. We also know with recent clinical trial data that women might actually respond differently to the novel therapeutic treatments that we have at the moment for Alzheimer's disease. The recent treatments, lecanemab and donanemab, as well as aducanumab, they all have some element of perhaps sex differences, with women perhaps presenting with a slightly less therapeutic response, at least to some of these anti-amyloid therapeutics. Much of this remains to be, further understood and explored. But at the very least, on one example of the Clarity AD trial with lecanemab, which is now Leqembi, they found that men showed a 43% treatment effect, while women only showed a 12% treatment effect. And so, because of this, it's not just simply capturing a snapshot of the community at the time, it's also understanding how treatment effects may be significantly impacted by sex. And when we think about what might be driving these sorts of differences between men and women in their risk for Alzheimer's disease dementia, one of the most enduring findings that has been known now for many decades is that the sexes differ in their level of risk of dementia based on their genetic risk for the sporadic Alzheimer's disease case. The ApoE4 allele is one of the most well-known risk factors, the genetic risk factors for dementia in older adults. And what has been shown time and again is that women who carry the E4 allele are at much greater risk for dementia than men. And that is particularly true between the ages of 65 and 75, as is shown in this particular figure that I'm presenting in front of you. If we think of the duration of a dementia diagnosis from the time of sort of showing this earliest stage of pathology in the brain all the way to a point of a diagnosis, it's actually quite a long duration. It's about 20 years. And if we take this 70-year mark that I've shown here on the screen of the difference between men and women showing rates of incidence to dementia, if we take that back 20 years, that takes you to the age of about 50, when the genesis of Alzheimer's disease is really starting to occur. And around the age of 50 is when women are starting to go through menopause. And so this is sort of, I just wanted to initiate that thought in your mind about sort of hormones and menopause when thinking about these sex differences in relation to risk for dementia. But it's also not just about the clinical diagnosis. We also think about what's actually going on inside the brain in terms of pathology. What you see in front of you is two bar plots. On the left-hand side, you're seeing the density of neurofibrillary tau tangles. This is one of the hallmark pathologies of Alzheimer's disease in the brain. On the left-hand side, you're seeing the density of these tangles in the brain in men, differentiated by age. And on the right-hand side, you're seeing the same kind of bar plot, but for women at each decile of age. Each of the different colored bands is indicating where these neurofibrillary tangles are being located. The first thing you can immediately see just upon visualization is that women are showing much higher amount of these tangles, these tau tangles in the brain than men. This is particularly true both at the lower ages between 50 and 70 and at the upper ages, 80 and above. In the older ages, this makes a lot of sense. Women tend to live longer than men. But at the younger ages, this is what's particularly interesting to us, because again, it's sort of highlighting this incredible age range between 50 and 70, which I just highlighted in the previous slide, as sort of this initiation, this sparking of when sex differences might appear. And again, sort of implicates this age range of when menopause is starting to take hold in women. Now, I wanted to sort of talk about this idea of risk and resilience, because even though we think of women being at greater risk of dementia, there is also a very strong story to tell about women also showing resilience. And I think this is a really important story to tell, and is actually much more interesting in some ways, because it can really shed light on where we think there might be nuggets of gold and discoveries that are yet to be made about harnessing the superpower of women and being able to really optimize brain health, particularly for women. So what we are showing you here, or what I'm showing you here, is a bar plot, or two sets of bar plots. On the left-hand side, there's 4 bar plots that are associated with healthy controls, and on the right-hand side is for symptomatic patients. The blue box plots are of those people who have no evidence or very, very low levels of pathology in cerebrospinal fluid. And when I talk about pathology in this case, I'm talking about the 2 most common pathologies found in Alzheimer's disease, amyloid and tau. In the red box plots, you're seeing those people who we assume have very high levels of pathology based on the cerebrospinal fluid measurement. And what you can see on the very left-hand side is that men are showing much worse performance, so higher number in this case on the y-axis is worse performance on a memory task. you're seeing that men are not doing so well in when they're healthy and they have higher levels of pathology in the cerebrospinal fluid, but women are actually doing very well. Regardless of whether or not they have high levels of pathology, they're actually performing equally well and actually better than any of the male groups. On the right hand side, once you get to a symptomatic patient group, however, men are still doing more poorly if they have evidence of these biomarkers. But what is really striking is that at the point of a diagnosis of dementia, women really start to show a much sort of more, I guess, compromised outset because of having this elevated level of pathology. And so I think what this sort of cross-sectional study shows or is starting to give evidence towards is that there is a high amount of resilience for women early on in the disease. They can really bear the brunt of pathology quite well. But once we get to a diagnosis of dementia, there seems to be this sort of loss of integrity over time. So what we wanted to do was to see if we could visualize this over time. Cross-sectional studies are fantastic, but what you really want is to be able to sort of visualize this longitudinally. And what you see in front of you is a study of about 1,000 older adults. And what we did was we measured their cognition over about 6 years. And we wanted to see whether women and men differed in their cognitive slope over time. On the y-axis, what you're seeing is a composite of cognition with a lower score, meaning poorer cognitive performance. And what we did was we divided men and women into their level of risk for Alzheimer's disease. They either had high or low levels of amyloid in the brain based on neuroimaging, or they were a carrier of the genetic E4 allele that I mentioned earlier, or they weren't. And what we found, and what I really want to focus on, is the red and the green lines in front of you, the bolded lines. The red line is women who have both of those risk factors. They are E4 carriers and they have high amyloid. The men are in the green who also have the same level of risk. What you can see at the beginning, the cross section, is that women were performing much higher than men, even with that level of risk that they had at the outset. But what you can see is traversing the study, women show this huge waterfall effect. They sort of really show this rapid decline in their ability to function on this cognitive composite. And men actually perform relatively well over time. And this is, I think, pretty interesting evidence towards a potential loss of resilience in women over time. Now, I've talked a little bit about, the global sort of prevalence rates. I've talked a little bit about pathology in the brain, looking at tau tangles in the brain. And I've talked a little bit about resilience. But one thing I haven't really mentioned is we might see these tau tangle differences in post-mortem tissue, but can we see this in real life in the brain in younger women? And when I say younger, I'm talking about, 70s, because a lot of the work that we've looked at in the past when thinking about post-mortem differences in tau tangles, we're talking about older adults that are much, much older. They're in their 80s or 90s. Here, this is one of the first results that we ever showed, showing that women have much different levels, much higher levels of tau in the brain based on neuroimaging. So what you're seeing in front of you is actually anything from a blue color all the way to a red color is showing much higher levels of tau signal on PET neuroimaging relative to men. We were quite surprised by this finding because we were assuming we might just find higher levels of tau in the areas of the brain that are most impacted by Alzheimer's disease. Those tend to be regions of the brain that are associated with memory. If you think of a baseball glove, a baseball mitt, it's the sort of the thumb area of the mitt. That's where you're usually seeing the earliest signs of Alzheimer's disease, particularly for tau starting to appear. And indeed, if you look in that baseball glove area of the mitt on the bottom, just on the right, bottom right as an example, you can see there's a very hot color, a red hot color that is showing much higher levels of tau in women than men. That is actually what we would have assumed to see. But you can also see actually proliferated across a lot of areas of the brain, including the neocortex, so in the outer areas of the brain, there are much higher levels of tau. What I'm not going to show you today, but what has been shown many, many, many times over and over and over again, is that we don't actually see differences in the levels of amyloid between men and women. So there's an interesting dichotomy that there's these two hallmark pathologies of Alzheimer's disease, that amyloid doesn't seem to really show differences in their levels between men and women. But once you get to a level of tau, it really starts to show a difference across the board. And so what does this higher level of tau in women impact over time? Well, I would argue that it impacts faster rates of decline in cognition in women relative to men. What you see in front of you is a series of spaghetti plots. Each of these little tiny lines is indicating a real person's cognitive function over a period of time, about 10 years in a study. I've divided these spaghetti plots into three based on how much tau they had in their brain at the time of their first tau scan, which actually was in the middle of the study. So that's why that 0 point is sort of in the middle of those spaghetti lines. The blue lines indicate men and the red lines indicate women. Now, in the lower levels of tau, the lower tertiles, which is the first two panels, there really isn't a difference between men and women. And if anything, actually, women do still perform a lot better than men at the baseline. But what I've done here is I've made the baseline equal between men and women. What you find on the final panel is that women start to really accelerate in their decline if they have equally high levels of tau as their male counterparts. And so to me, this again demonstrates that there's sort of this potential vulnerability to tau, which is having then an impact in rates of decline and then having an impact on this sort of potential progression to dementia. And we're not just seeing at the cross section at a single time point that women have higher levels of tau, but that also they show an acceleration of tau in the brain. So what I'm showing you here is work from Dr. Gillian Coughlin, one of the incredible postdocs in my lab. And what she's showing here across multiple different cohorts is that once women have higher levels of amyloid, which is what we call, abnormal or PIB positive on any one of these panels, it's the right-hand panel, you can see this huge increase in the level of tau, which is on the y-axis, in women versus men. What this is telling us is that there is a way to actually visualize the point at which women do start to have higher levels of tau than men, and it seems to be happening once they already have abnormal levels of amyloid. So as I said to you before, there doesn't seem to be a difference in levels of amyloid in amyloid PET imaging. There does seem to be a difference in levels of tau, and this seems to be very much tied to once you've already hit a level of amyloid burden in the brain. But again, this is not all a doom and gloom story. There are actually many instances, even in once women already have a level of tau burden in the brain, that they may already show much better outcomes than men. This is one example from Rick Ossenkoppela's lab in Amsterdam, showing that in certain regions of the brain, these are highlighted in purple here, for a given level of tau in these particular regions of the brain, women actually have what I would call juicier cortices. So they might have a high level of tau there, but women have still thicker cortices in those regions. So more volume in that sort of cortical layer of the brain relative to men. One argument could be they are able to hold on to that level of pathology and still maintain brain health as well as cognitive health in the face of a high pathological burden, at least for a period of time. So, what is the implication then of this sort of resilience in women? One thing that we know, and I sort of alluded to earlier on in the talk, is that women tend to outperform men on many tests of cognition, memory, and thinking. That's not all tests, and I don't want to disparage the men out there watching this talk, but women do perform a lot better, particularly on verbal memory tasks. And on most of the diagnostic batteries that we use to diagnose dementia currently, many of those diagnostic batteries include verbal memory tasks. Because of this, women may in fact be underdiagnosed for dementia relative to men because they perform so much better. So there was an incredible study done by Erin Sunderman and colleagues. They're based at UCSD. This was a few years ago, but they have replicated this work time and again, showing that if we actually adjusted people's test scores based on women's norms or men's norms, we can actually better diagnose women. And what you're seeing in front of you here is survival curves. So people progressing to dementia on the sort of bold black line at the very bottom showing the worst survival. Those are women who are true positives. They were accurately diagnosed early on in the study and then did progress to the dementia. In the top sort of very dotted line, those are the true negatives. Those were the women who were clearly performing well, and they were much less likely to be progressing to dementia. The line in the very middle, these are who we consider to be true negatives. These are, false negatives. These are women who were told or diagnosed based on their performance that no, you're doing fine, you don't need to worry at all. But in fact, they were underdiagnosed because their norms were sort of based more along the lines of men and women combined. And in fact, these false negatives are much more likely in women than men. So it's a point to bear in mind. So this is my sort of idea of the hypothetical progression of Alzheimer's disease by sex. We don't really think there are differences in amyloid load between the sexes, but once you hit an abnormal level of amyloid, women really split apart from men in their proliferation of tau, which we believe then results in faster rates of cognitive decline and potentially higher level of risk to progression of dementia. But let's get to the really fun bits. Let's talk about the debate of, is it hormones or is it genes? And I was going to do a poll about what you might think the answer would be, but I felt that it might be a little bit patronizing because, as we all know, there's a bit of both. But I wanted to present elements of both sides of the story because really it's very hard to mesh the two together in some cases, although there are some beautiful studies of this and I'll refer to them later on in the talk. But let's start out with hormones. So just as a primer for everyone out there, we see a lot of changes in hormone levels as we age. Particularly for women around the age of 50, there is a drastic and significant drop in estrogens and progesterones. With men also showing a slow decline, a slow but steady, more linear decline in testosterone levels. And going to sort of the right-hand side panel here, as we age, while testosterone is declining, this is evidence in women and in men, actually, in women as well, this can be found. We also see something that increases over time, which is called sex hormone binding globulin. And I won't touch too much upon that here in the talk today, but there is also some interesting evidence of sex hormone binding globulin potentially having a role to play in sex differences for AD risk. One of the simplest things that we can look at is menopause status and how that might be associated with Alzheimer's disease risk. And here, what this particular study was, Lisa Mosconi's group, was really looking at, can we look at amyloid burden in the brains of older adults, women in particular, and can we then capture women based on their menopause status at the time? Are they asymptomatic and perimenopausal, so they're going during, they're through the menopausal period at the moment? Perimenopausal, meaning they have symptoms and they're going through menopause, or are they entirely menopausal and relative to their age-matched counterparts, male counterparts? And what you can see here in this particular figure, which is quite interesting, is that women at this sort of perimenopausal phase, please note that these are much younger groups of women than I have showed you so far, they're showing higher levels of amyloid. So there might actually be a small story to play for amyloid load playing a role in differentiating men and women, maybe during menopause. We've extended these findings ourselves, and we're looking at menopause status in terms of tau here. And what I wanted to look at was menopause and premenopause women relative to age-matched men. And what you can see here is on the very right-hand side, this is sort of a violin plot of women who are postmenopausal. They have much higher levels of tau than their age-matched male counterparts, as well as the premenopausal men and women. So again, showing that even at that much earlier age of about 50 to 60, we're starting to see these differences in these differences in AD biomarkers emerging. Now, my postdoc, Dr. Coughlin, took it one step even further, which is such a fantastic study. This one, I love it, where she wanted to not just look at status, like are you postmenopausal or premenopausal, but she wanted to look at the age at which women go through menopause. The idea is that the earlier age at menopause might actually be associated with a earlier being removed from estrogens earlier in age. Estrogen really is a superpower of the body and the brain for women, and having that sort of receding level of estrogen so early in life can potentially increase risk. And it's certainly been found in quite a lot of observational studies of other outcomes and in animal models. And what Gillian found is that for a given level of high amyloid load, there was much higher levels of tau in women who had early menopause onset. So we're talking about women who were, earlier than 40 at their age of onset. There weren't as many of those women. It is a lot more rare. But we did see this very interesting difference between those women and other women who had much later onset. In addition, and counterintuitively, we also found that women who reported using hormone therapy at some point in their life had higher levels of tau as well. And we found this a big head scratcher, because you'd think that those women who had earlier age at menopause, you'd think that that would mean that taking hormone therapy to try to account for that or compensate for that should actually increase your protection. But in fact, we found the opposite. Gillian didn't just let that rest, though. She did some more calculations. And one thing that we have known in the literature for a long time now is that the timing at which you take hormone therapy matters a lot. The Women's Hormone, the Women's Health Initiative is one of the largest clinical trials that's ever been run in the world, but it was on hormone therapy. They had to halt that trial early because women who took hormone therapy in that trial actually had more than twofold risk of dementia, in addition to a whole bunch of other outcomes as well. And one of the things that they found, or one of the things that was a part of that trial, was women who were taking hormone therapy in that trial initiated it at the age of 65. That's about 15 years after going through menopause. So what Gillian decided to do was then split people in this study based on how far away they had initiated hormone therapy from when their menopause onset was. And lo and behold, we found, similar to the Women's Health Initiative, that those women in the yellow group who initiated hormone therapy much later in life, these were the ones who were driving the higher levels of tau, not the women who were taking hormone therapy or initiating it similar in time to their menopause onset. But let's turn for a second to another part of the story, which is the genes. Because at the end of the day, I don't think the hormones work in isolation. There is such a rich story to tell of the X chromosome, and we really are only just scratching the surface of this area of the field. And one of the reasons for that is because the X chromosome is in fact extremely difficult to measure and study. But let's start out first with the powerful story of being a woman as a superpower. So women live longer. This has been known for a long time, and this is not just in the general sort of world or higher OECD countries. This is also known across animal species as well. Women also live longer with dementia. So what you can see on the right-hand side here is the blue line is indicating the life expectancy of women who are AD patients versus the green line, which is men who are AD patients. Women have a much longer lifespan even with dementia than men, showing that there is a powerful story to play for resilience in this thought of AD. And what might be driving some of this? Well, there's an incredible story to tell about the beauty of the XX chromosomes. On the left-hand side here, this is a beautiful demonstration of what the XX, or what a heterogamete or a homogamete how they might differ in their longevity. On the left-hand side of those dots, there's a line going through, and on the left-hand side, that's showing you homogametic sexes dying earlier. When we're talking about homogametes, that is a doubling or a copying of the same sex chromosome. In the human example, that's XX. On the right-hand side is heterogametes dying earlier. In the human example, that's the XY. But you can see across many species, not just humans, that the heterogamete dies earlier. And you can see there's blue dots and red dots here. In the animal kingdom, there's some birds, there's some lizards, there's some butterflies that are female heterogametes, and they are dying earlier. The argument being that there's something about that second copy of the chromosome rather than the gonads that might be having an impact on longevity. On the right-hand side here is this beautiful design that has been set up quite a long time ago by Dr. Art Arnold, where you can actually split apart in an animal model the gonads from the sex chromosomes simply by taking the SRY gene that can be flicked on and off to turn on or grow the testes or not. You can move that SRY gene to the autosome and then manipulate it yourself so that you end up with this beautiful two by two contingency table of a female rodent with XX, with the ovaries that is XX or XY, and testes that is XX or XY. And using this particular 4-core genotype model, Dr. Dina Jubal and her lab based at UCSF did the most incredible study. And this is one of those inspiring moments where you realize, my gosh, science is so cool. They took an animal model of Alzheimer's disease, the amyloid precursor protein mouse, which proliferates amyloid through the brain very early in a mouse's life. And they paired that with the 4-core genotype model. Long and short of it, they found there was a very strong impact of both gonads and sex chromosomes to impact survival rates. So you can see that on the very right-hand side, that the purple line, which is those XYs who had the testes, had a really poor survival outcome. But the XYs who had the who did not have the testes, who had the ovaries, had a really bad outcome. But the thing that I really wanted to point out here is it wasn't the gonads that were doing all the work. It was really the sex chromosomes that were having an impact. They took that one step further on the right-hand side to ask the question, is it about the Y chromosome specifically, or is it about the X? And what they did was they paired lots of different mice models together to end up with, and this is the most important component, the X0. So the X0 was just one X and nothing else. No Y was paired with it, or any other mouse model, which had X and Y all associated. And what they found was the mouse that had the poorest survival was the one that was X alone and nothing else to compensate, really arguing for the idea that the second X is really important for resilience. So we've come to the point in the story now where the XX is really fascinating, because in the XX, you have this overdosing problem, because men only get one X from their mum and that's it. And the women get one X from their mum, one X from their dad. In order to avoid these genes being overdosed, one of the X's across every single cell in the body gets randomly silenced for women. But that silencing mechanism is not complete, which is fascinating. Some of the genes characteristically tend to escape inactivation. A lot of these genes, interestingly, tend to be involved with inflammatory diseases and sort of neuroinflammatory responses as well. This is one of the arguments for why women are more at risk of autoimmune disorders than men. What Dina Jubal's lab then did in the same study was to then understand how is this X inactivation playing a role for increasing risk or resilience. And what they found, what you can see here in front of you, is the X chromosome gene expression in brain tissue associating with cognitive decline patterns in women prior to death. And what they find is in the blue lines that are coming off it, there's lots of genes that are associated with protection against cognitive decline, but also orange lines indicating risk, that there is greater sort of greater levels of expression of these genes, these X-linked genes in the brain, might be associated with faster cognitive decline in women. We wanted to take this a step further. This is my other postdoc, Dr. Mabel Seto, who wanted to look at a lot of different outcomes in a similar sample. And we wanted to look at amyloid and tau, as well as cognitive decline, to see how some of these genes might repeat across different outcomes, and how they might show us something about risk and resilience in women based on the X chromosomes. Here, what we find for amyloid plaques and tau tangles is that there are quite a few resilience genes and a few risk genes. Interestingly, these genes tend to repeat over and over again. Not all of them, but some of them really do repeat over and over again across these different outcomes, suggesting that there may be targets of interest that we might want to pursue further in future to see if they might be drug targets. We then again went further and to see how these genes might again repeat in measures of cognition. And again, these are really interesting sort of areas to explore further. So that was on the X chromosome. But the final piece, which I think is really important, is to not ignore the autosome. There are also sex differences in Alzheimer's disease risk based on the autosome. This is work that is done by our collaborators at Vanderbilt. Dr. Logan Dumitrescu is leading that lab. And what they were interested in looking at is how autosomal gene expression might be associated with our outcomes of interest, the same outcomes, amyloid, tau, and cognition, how they may be associated differentially by sex. So we already know in those green colors in that circus plot, you can see in the green colors F, P, and C. These are different brain regions, but these are all the genes that are associated with amyloid, tau, and cognition that are shared between the sexes. There is no difference. The little tiny part of that pie, I guess you could call it a pie, you can see sort of red to orange colors and blue to purple colors. Those are female specific associations and male specific associations. The most interesting part of this finding is they found that across the entire autosome, about 9% of the associations with amyloid, tau, and cognition are sex specific. 9% doesn't sound like a lot, but when you think about the sheer number of genes, 25,000 are sex specific, that really makes you stop and think. In addition to that, they found that this sex specificity was really slanted towards women. There were 3.5 times more likely to be female specific associations on the autosome than males. And some of these are highlighted here. We won't delve too deeply. They're working on a publication now with Dr. Seto, and that should be coming out soon. I'm very excited about it. But there are definitely a lot of genes associated with both resilience on the autosome and risk, which are very female slanted. And I think that really deserves more understanding and further exploration. So I guess I wanted to just sort of round out this particular section to highlight the importance of understanding that genes and hormones together are incredibly important for understanding risk for Alzheimer's disease, particularly when we're thinking about pathology, but also thinking about incidence to dementia. There are many things or many elements that genes and hormones can impact that link between the AD pathology directly and the gene and the hormone. So for instance, they can impact metabolic syndrome, disrupted sleep, poor mood, and cardiovascular risk. All of those elements, which are really huge elements for Alzheimer's disease risk, they are impacted directly by menopause. Hormone therapy use is a really important area that we still need to explore further. But as well as we know that the immune system and neuroinflammation in particular is a very strong driver of Alzheimer's disease pathology. And we know there's a link with the X chromosome. So there's a lot of interplay going on here, and further sort of research needs to be done. But I think that it really sort of highlights for us how complicated and nuanced this picture is. I wanted to sort of highlight very briefly an example of how all of these elements can sort of tie together. In this particular example, this is Dr. Caitlin Casaletto's work from UCSF, showing the inflammatory pathways that might differ between the sexes to impact amyloid and tau levels in post-mortem tissue. So this is a very, very large study. So they had about, I don't know, 400 individuals in both the female and the male groups. And what they were interested in looking at was the amount of microglial activation in surrounding brain tissue relative to the amount of amyloid plaques and tau tangle burden. What they found in the female sample was that there was a feedback loop between microglial activation and amyloid, that more microglial activation predicted more amyloid and vice versa. And each of those had an impact on tau. For males, on the other hand, there was no feedback interaction between the two, microglial activation and amyloid, but both had an independent and totally, a sort of orthogonal association with tau, suggesting that even though men and women may experience dementia similarly, they may have similar rates of incidence potentially, but the pathways to get them there are entirely different. So that is a really fascinating study. So what still needs to be addressed? So many things. And I can't wait. I hope there's many people in the audience who are fired up and ready to go and want to join us in this fight to understand sex differences in Alzheimer's disease, particularly from a women's health standpoint. There are many things. Survival and comorbidity is a really big problem to try to address. Women live longer than men. It is possible that what I have showed you today, women having higher levels of tau, for instance, could simply be because women are able to hold on to the tau for longer. The story of resilience is a really powerful one, but it makes it really tricky to fully understand, do women have a vulnerability to tau, or is it that men simply do not survive in the study for long enough to show us higher levels of tau? Have they progressed to dementia already? Have they passed away? Do they have some other complications that make it difficult to assess? That's where comorbidities comes in. There are different levels of vascular burden at older ages for men and women as well. So we need to take into account all of these different nuances. We also don't fully understand why women are at greater risk if they carry the E4 allele. There are some suggestions of what that might be, but it hasn't really borne out yet in the literature or in research. So that's an area of hot sort of focus right now. We don't really understand how inflammation plays a role. I think understanding more about the X chromosome is going to be vital for this. Whether or not females are resilient or susceptible is another key thing to understand. I think a lot of the literature focuses on susceptibility, and I think that is a real downfall in some cases, because I think we're pathologizing something that doesn't need to be. And in fact, there's many sort of nuggets of discovery that we can make with relation to drug discovery, thinking about resilience in women. In addition, there are intersections that we cannot ignore about gender and the sociocultural sort of differences between what it's considered to be a male or a man and what's considered to be a woman that can have a huge impact on these outcomes, as well as race and ethnicity. And I really did not give any sort of focus on that topic. It deserves its own talk. But I wanted to thank you all for considering what we have been working on for many years now in my lab and many other labs across the US and the rest of the globe. I'd like to give special thanks to my co-authors and collaborators and colleagues and friends, as well as the Harvard Aging Brain Study Lab, my funding sources, as well as my beautiful daughters, who I hope one day will grow up in a world where they do not need to worry about elevated Alzheimer's disease risk. And I'll take questions.
Thank you so much, Rachel. What a wonderful presentation that was. Just before we do jump into the Q&A, I'm going to give you a second if you need to catch your breath or maybe sip your tea or water. And I just want to remind the audience that the Q&A section is there for you to send in all of your questions. We are going to try to get to as many as we can, but we have already received a lot. So just bear in mind that we will try to get to as many as we can. I also want to quickly leave this pop-out link for you. If you have to leave early, please just submit your feedback, your thoughts, comments. You can also submit a question there if you do have to leave early. And we really appreciate your feedback on your experience today. Okay, let's not delay any further. Rachel, I'll bring you back in now. Hi again.
Hi.
Okay, yeah, like I said, we have so many questions already, so let's get
Wonderful.
right into it. The first one for you today is from Samantha. Has there been investigation into women who have used hormonal contraceptives which reduce endogenous estrogen levels on tau levels?
Yeah, it's a wonderful question. I get this one quite often. There has been some work looking at oral contraceptive use on incidence of dementia. That's been done in studies like the UK Biobank, which is an incredibly large study, a more epidemiology study based in the UK. And there has been some other work as well looking at incidence rates also in University of California, Davis. Both of those studies and any others that I'm thinking of have been somewhat mixed in their results in terms of what oral contraceptive does for later life risk. The rationale and the issues involved with understanding oral contraceptives is multifaceted, but there's two major issues. One is the life course issue. You're taking oral contraceptive much, much, much earlier in life for a period of time that is very variable amongst women. And then there's an intervening period of many decades prior to then a diagnosis of dementia, which makes it very hard to sort of adjust appropriately for all of the different life course components that can occur during life intervening. And the other issue is that, in fact, oral contraceptives are very, very different in formulation, dosage, how long you take them for. One example is taking the estrogen progestin combined formulation of oral contraceptive versus not. There's also the issue of taking whether it's oral or, in fact, you might be thinking of other types. There's the Nexplanon, for instance, which goes under the skin and releases over time simultaneously. But there has been no definitive work so far on oral contraceptive use and, for instance, amyloid and tau in the brain yet. My feeling at the moment is there may be some interesting stories to tell, but so far the literature has been somewhat unclear.
Okay, thank you so much for breaking that down for us. Our next question from Alex. I've read that women who have had a hysterectomy may have higher risk of developing dementia. Do you know the mechanism behind this, and do you know whether it would be related to hormones?
So Dr. Walter Rocker would be the perfect person to speak to this, and I wish he was on the call right now. And I strongly advise attending any of his talks. He is the one that's really been a big sort of leader in the field of looking at hysterectomy. In particular, we need to be very clear about our terminology here. We're not talking about removal of the uterus. We're talking about removal of the ovaries. That is the most important component when we're thinking about surgery. So it's actually bilateral oophorectomy that we think about. And yes, there is actually a very strong amount of literature suggesting that those women who have undergone a bilateral oophorectomy at some point in life, particularly earlier, are at much greater risk of cognitive decline than women who have not, or women who have just gone through what they call spontaneous menopause. They used to refer to it as natural, but that comes across very poorly, and we now call it spontaneous. In our hands, when we have been looking at our own observational data, our numbers are very low. We don't tend to have the statistical power to look at those women who have gone through spontaneous menopause versus those who have gone through some kind of surgical intervention, primarily bilateral oophorectomy. One of the issues is when you ask women what surgery they went through, some cannot, in fact, tell you whether it was just a hysterectomy or it was a hysterectomy and bilateral oophorectomy. So it gets really hard and complicated trying to tease that apart. At the very least, those studies who have done it very, very well have been able to see something. In our hands, we have not tended to find a greater risk of surgical intervention on risk for higher levels of tau or amyloid. But that remains to be seen. And we do want to look at this further going forward. It's a great question.
That's great. Hopefully, yeah, you have an answer for us soon on that. um, okay, our next question from Brittany. For the studies noting the relationship between early menopause and HRT, is the earlier menopause caused by surgical/chemical intervention, or did these women just have an earlier menopause via natural hormonal changes?
It's a wonderful question. And again, we were asked this a lot by the reviewers in our paper, and we did try to explore this. Again, our numbers were too low to be able to fully dive deeply into this. But the women who were going through earlier age at menopause in that particular study did not have a greater propensity for surgical intervention. In saying that, it is more common, at least more prevalent in certain sort of social strata to have earlier age at menopause be associated with a surgical intervention. But there is also other strong sort of sociological factors that can drive earlier age at menopause, such as smoking and BMI. They have quite a strong driver or impact on earlier age at menopause. Another thing that I wanted to highlight, although this is not directly the question, but I think is really interesting, is that earlier age at menopause is sort of contextualized. It depends on what country you're coming from. So in the US, average age at menopause is about 50 or 51. But if you go to India or Latin America, the average age is about 45 to 46. So we really need to sort of think about what we're talking about when we're talking about what is considered to be early, and it's very contextualized and country specific and culture specific in some cases.
Wow, that's really interesting. I didn't know that.
Yeah.
Okay, Junie asks, we know that hypertensive pregnancy history increases risk of vascular dementia and mortality from Alzheimer's disease. I'm curious as to whether all the studies described here considered pregnancy history. Perhaps those with accelerated tau pathology had history of hypertension/HPD.
I would really love to welcome you to our sex and gender professional interest area for Alzheimer's disease. If anybody's interested, you can find me on LinkedIn and I can direct you to it. One of our working groups is to look at this very question. What is the impact of adverse pregnancy outcomes on Alzheimer's disease markers, whether it be amyloid and tau, or whether it be cognitive decline or dementia incidence? There is certainly some very interesting evidence, at least at the epidemiological level that you referred to, that suggests that adverse pregnancy outcomes, particularly hypertension and preeclampsia, even preclinical elements of preeclampsia, has some very potential negative effects on incidence rates. But there really is not very much known about amyloid and tau. There's a very good reason for that. And actually, there's a very good reason for many of the things that we haven't studied yet. And one of those, the reason is because we don't ask. Many of these observational studies do not gather information on pregnancy or oral contraceptive use, and they don't even ask about menopause. So we are very limited in what we can study at the moment, which is why I am on a big sort of push to try to get more new cohorts out there that will ask these questions. And if you're interested in hearing about that more, there's a big concerted effort in the Anne S. Bowers Women's Brain Health Initiative to really address this, this gap in the literature at the moment.
Amazing. It does seem like there's a few gaps that
There are a few, but
that
you know
we'll need
what?
to go for.
The wonderful thing about that right now is that it's low hanging fruit. We have discoveries that we could make, massive, high impact discoveries just around the corner. We just need to start looking, and that's exciting.
And that's why a collaboration is so wonderful. So I
Couldn't
love
agree
how
more.
you, told people to reach out.
Yes.
Amazing. Um, Richard's question asks, stress disrupts sleep and is a significant risk factor for neurological disease. Have
Mhm.
you considered how stressful experiences at various life stages can influence income outcomes?
That's a really good question too. No, it's not something I've looked at, but certainly, I think of stress in multiple ways, whether it be, sort of biological cortisol levels, or whether you're thinking sort of life stresses in a sort of psychosocial, psychiatric level. In both levels, I am very, very not well versed, but I would be more than happy to put you in touch with colleagues of mine who do some of that work. From what little I do know, there is actually impact of stress, and certainly stress increases a lot biologically during the menopause transition and does impact a lot of things. And one of the arguments is there may be an impact on HPA axis during vasomotor symptom experiences. So there's a lot of sort of biological effects, but there certainly as well are sort of elements in the environment as well that should be having an impact as well. Unfortunately, it's not the area that I do. Only so many hours in a day.
Yeah, unfortunately.
Yeah, that's right.
Okay, thank you for that. Um, the next question is from Lisa. Are hormonal contraceptives in premenopausal women protective of dementia long term?
Yeah, this is a question I get asked a lot actually in my sort of community talks. A lot of women who come up and go, "Should I be taking hormone therapy or should I not be taking hormone therapy?" And actually, it's a really good question. What we do know from the guidelines of the American Menopause Society and the International Menopause Society, there's two of them, and they have yearly meetings. They're fantastic. I highly recommend. They both have guidelines recommending that women should take hormone therapy around the time of onset of menopause if they're having significant symptoms. There is only really one, proper way to treat, or one robust way to treat vasomotor symptoms, which are so horrible to go through, and that is with hormone therapy. There is also guidelines to say that there is a certain period of time, a duration that women should, be able to take it and still be healthy. It's around a decade. So, and maybe up to 15 years, depending on speaking with a specialist. And I certainly am not a menopause doctor or an endocrinologist, so I cannot provide guidance in that sphere. But I would say that there are certainly guidelines, and people are absolutely able to take hormone therapy at a certain time of life. But I certainly wouldn't, recommend necessarily initiating it later in life uh, for the purposes of trying to reduce levels of tau.
Okay, great. Thank you. Um, we do still have a lot of questions, so if you have a couple extra minutes to
I
go.
do, I
Okay.
do. If everybody else does, I don't know if everyone can say whether they have time, but I
Yeah.
do.
Okay, great. Yeah, for the audience, if we don't, if you have to jump off, the recording will be available soon later today. So the questions that we address, if you once you leave, will be answered in there. And I'm going to
Mhm.
pass them all to Dr. Buckley as well after the event. So hopefully you can stick around though. These are great questions.
Yeah,
So
fantastic questions.
Luther asks, if women are showing less treatment effect in trials but worse pathology at death, does that indicate that trials need to be designed to address this consideration?
I am I am actually a big voice about clinical trials and sex differences in Alzheimer's disease clinical trials, and you can see some of my editorials out there if you're interested in my opinions. My feeling at the moment is it's too early to say that we need to change clinical trials or how therapeutic sort of study designs should be set up. But I do think we are at the intersection where we need to start thinking about reporting and clearly and effectively and consistently reporting sex differences or sex specific outcomes in therapeutics so that we can better understand how treatments are impacting women and men differently, if they are at all, because there are some indications that there may not be, or if they are, to what extent they might be, or in which context. So for instance, the lecanemab result that I talked about before, that showed 42% effect in men and only 12% in women, or 43 and 12, that actually was also shown to be a huge difference in whether you were a carrier or not a carrier of E4. And we know that there is a sex by E4 effect. So it's things like this, thinking, keeping the literature in mind, and being really held accountable to report all sex differences and what those results are at every single stage so that we can better understand how these things are happening. I do think we can learn a lot more as we go. Whether or not we're at the stage where we have to run totally different trials by sex, I don't think we're there yet, but we do need to be more open about reporting.
Okay, excellent. Um, Kumika's question says, in general, women's life expectancy is longer than men. How
Mm.
is it considered in the analysis of the longitudinal outcome?
Yeah, this is a huge question we get from epidemiologists, and I'm going to tell you right now that we are at a huge, I guess, where we're really not doing due diligence there. A lot of our observational studies, when we run our models, we do not account for longevity. It is a very difficult type of calculation to run. I have seen incredible models by Dr. Elizabeth Rose Maeda's lab who try to simulate a survival effect, so how long people might live for and how that might impact these models. And we really should be doing more to try to include those types of elements in our models. But no, we do not do that at the outset. One of the things that we did do in our mortality rate finding early on in the talk was that we did adjust for age of death. And even after accounting for that and other comorbidities, women still died more with dementia.
Okay. Yeah, makes sense. Thank you. Um, Maurice's question is,
Mm.
when you refer to tau, are you always referring to hyperphosphorylated tau? Can you just
Yes,
clarify?
it's a great question. Yes, we are. It depends though, because we're starting to sort of get into the realm of looking at plasma markers of tau. So we are often, when we're looking at the neuropathology version of tau, we're thinking about tau tangles, neurofibrillary tau tangles. When we're talking about tau PET, which is the neuroimaging data that I showed you, we're talking about sort of the more tau tangle, hyperphosphorylated tau. And then finally, when we're thinking about the plasma and cerebrospinal fluid, that is what we're hoping we're looking at, is the hyperphosphorylated version of the taus. But still, lots to be understood and explored when we're thinking about plasma.
Yeah, okay, excellent. Um, I think this will be the last question that we ask. Yeah, answer it however long or quickly you'd like. Are there known correlations between hormone depletion diseases such as hypothyroidism and
Mm.
Alzheimer's disease risk in women?
Mm.
From
Yeah.
Emily, thank you.
Yeah, that's a great question, and I'm going to be short on this one because we haven't looked into it. But what I do know is there are some groups that are quite interested in it. There's huge power issues again when we're trying to think about these things. Although that is not, it is quite a common disorder. It is still, if you're then splitting into just a female group and then looking into sort of hypothyroidism and things like that, then you do end up with real power issues, at least when you're thinking about neuroimaging. But at a population level, at an epidemiological level, there are people interested and there seems to be some kind of signal there, but it's not an area that I'm familiar with. It's a great question.
Okay. Yeah, thank you so much. You did a great job answering all of those, even ones that, like this one that you haven't necessarily looked into yet. So I just want to say thank you so much for being here and, being our first event in the Women's Health series for this year. Your passion really shined through during your talk, and it was so interesting. So I hope you had fun today.
It was a pleasure, Sydney. I really appreciate it, and it was so much fun. Thanks for everyone to attend.
Yeah, fantastic. Okay, so just to close out, I want to thank our live audience. We know you have busy schedules, so we really do appreciate you being here live and even sticking around a little late. And I hope you enjoyed your experience as well. Please join us for the next event in the series, which is coming up on Wednesday, October 16th at 11 a.m. Eastern Time. You will receive a reminder email with the link to log in soon. Make sure you block off your calendar so you don't miss out. A quick reminder, a recording of today's webinar will be available soon, so keep an eye out for the email that will give you access to that. Additionally, that survey will appear at the end of the webinar. We do read all of your comments and we appreciate your input on your experience today. You can also let us know what webinar topics you'd like to see in the future. Finally, if you still have questions, you can continue to submit them in the survey and we'll be sure to pass them along to Rachel. In closing, we hope you enjoyed this Inside Scientific webinar. Another quick thank you to our partners and sponsors for helping to make this series possible, and I hope you all enjoy the rest of your day.